Welcome back. In the last session, we have revisited the processes of involved in the central dogma of biology, that means the flow of information from DNA to proteins and the processes, mechanism of various processes, mechanism of various enzymes have been discussed. Okay. Now, we will discuss the amplification of a piece of DNA, that if we have a small amount of DNA, then how can we get thousands and thousands of copies of this DNA. Now, remember, copying of DNA in the biological system is called replication, that is the copying of DNA. But here we are talking about something which is carried outside the living system in a test tube or Eppendorf, but utilizing the utilizing the different components of the biological system that are involved in replication processes. We will show you how to do that, but this is important to distinguish between the two processes. Replication is the process which takes place inside the organism, living organism, okay. And that is what is called in vivo process. And the amplification that we are talking about is carried out outside the living system, and that will be called, or that is called, an in vitro studies. So, there are two processes, one is called in vivo, usually it is written in italics, or the other is in vitro. This is done inside the living system, this is outside the living system. Okay. So, our topic of discussion today is that if we have a copy of a double strand DNA, then how can we make, without going into the intricate machinery of the biological system, how can we make copies of this, identical copies of this piece of DNA. Okay. Now, this is what is also called, this process is called cloning. What is cloning? Cloning is basically making identical copy of anything, that if you have a writing in a piece of paper, and if you make a if you scan it or if you xerox it, you will get the clone of your original system. Okay. So, cloning is nothing but making or creation of identical copy or copies. Now, this can be applied to a cell also, that if you copy the cell, if you have one cell, particular cell, and you make copies of that cell, that is also cloning, but that is cloning of the cells. If you say that DNA cloning, if I say DNA cloning, then basically you are talking about that multiplying or amplification of a piece of DNA, that is what is called DNA cloning. So, we are talking about DNA cloning in this case. Now, there are usually two methods that are followed. Okay. One is called polymerase chain reaction or PCR based method, polymerase chain reaction, this is also called PCR. And the other is kind of you can call semi in vitro process, because at some point of time this second method, this is suppose 1 and this is 2, the second method utilizes at some point of time the cells in a living organism. So, it is kind of a semi in vitro analysis and or technique. So, this is basically utilizing the living system. So, one is called PCR and the other is utilizing a living system. We will discuss both. And what are the steps of this amplification? So, what you do first, suppose you have this piece of DNA, and what I want is to copy, suppose I want to copy this region only, I do not require the full copy of the DNA, but I want to copy the DNA of this region. Okay. So, what you have to do? First you have to, the steps are cutting the DNA at precise locations. So, you have to cut the DNA at precise locations, but the cutting should not involve the segment that you are interested to amplify. That is number one. This cutting is done by sequence specific endonuclease, because it is done inside the DNA strand. So, it is called endonuclease. Also they are called the restriction endonuclease or the restriction enzymes. Maybe in earlier discussion somewhere I mentioned about these restriction enzymes. Restriction enzymes are endonucleases which recognizes which recognize specific sequence, and the beauty of this sequence is that they are all palindromic sequence. That means, if you read from 5 prime to 3 n, whatever sequence you get, if you read the other strand from the same 5 prime to 3 prime end, you will get the same or identical sequence. Okay. So, that is called, that means, it has got a local C2 symmetry, that if you turn it upside down you will get the same thing, but remember this is local, you cannot turn the whole DNA this way and then get the same thing. It is just only a particular location. These type of palindromic sequences are present, and they are recognized by these restriction endonucleases, also called restriction enzymes. So, they are also basically necessarily they are molecular scissors. Okay. Now, what you do after you chop this off, what you need is a what is called a vector, means you have to something which carries this gene or carries this DNA, and then carries this DNA, and then ultimately that carrier plus this DNA of interest, that can be copied. Okay. So, this is not polymerase, we are talking about in general what is done, we are talking about the second one in the living, that means amplification in living system, we are talking about that in the beginning. So, you have the DNA, piece of DNA, this is your region of interest, you cut the DNA from the two sides using restriction endonuclease or the restriction enzymes, and then incorporate into a carrier DNA. It is a carrier in this case, I will talk about this carrier molecule, this carrier, and then the DNA is becomes an integrated part of this carrier, and then this carrier has a property which can be copied when that is put inside a bacterial cell. That means, if you have a bacterial cell and if you put this, suppose this is your carrier. So, if you can put this carrier molecule inside the bacterial cell, so when the bacterial cell grows, so you will have bacterial cell containing, each bacterial cell containing this this carrier DNA hybrid. Okay. This hybrid, or this is actually also called recombinant DNA, when you, because the carrier molecule is a DNA, is made up of DNA, and this is also DNA. So, this is what is called recombinant DNA. Okay. Again, I just quickly go through it, that first you cut the your DNA from the two sides without cutting the zone of interest, and then you attach it to the carrier DNA, which is by the way called a cloning vector. Okay. A vector is nothing but a delivery agent, like we have vectors for malaria. We know that what are the vectors of transmission of one organism, microorganism into the human body via the mosquito. So, the mosquito is the vector for malaria, encephalitis, all this. So, vector is nothing but it is a delivery agent. It takes up the thing that I need to deliver and then brings it to the bacteria, and they are capable of self replication, but not by itself. When it is put in the bacterial in a cell, it's suppose it's a bacterial cell, usually that is bacterial cell, and then when the bacterial cell is grows, then each cell, then the plasma, this is this cloning, this is now a recombinant DNA. This recombinant DNA also makes its own copy in different cells. And now, the next thing, if you want to isolate this piece of DNA, you have to chop the bacteria, the outside layer of the bacteria, take this recombinant DNA, and then utilize the same endonuclease to cut it off from the carrier molecule. So, I can now say that first you have the recombinant DNA containing your DNA of interest, put it inside a bacterial cell, grow, isolate the recombinant DNA, and then endonuclease will give you the DNA of interest. Copies of, because you will get multiple copies, because from each cell you will get this rDNA, and from the rDNA you will get the copies of the DNA of interest. So, that is the system which is usually utilized. Now, this has been largely replaced nowadays by, or a very quick technique. This is little bit slow technique. However, this has got one advantage, is that if you can put it in the bacterial cell, now this cell you can preserve, and whenever you need this recombinant DNA, then you can ferment the bacteria which will make copies, and then you can isolate the DNA. So, this is this can run, in theory it can run forever, because so long as you have the bacterial strain containing this recombinant DNA, then you can always get hold of the copies of the DNA that you want. Okay. And the other important factor is that if these copies of, if this DNA piece is ultimately, after transcription and then translation, it makes a particular protein which you are interested, then you will all the time you will get, you do not have to break the cells, because you are not interested in the DNA any longer. If you are interested only of the protein that this DNA makes, then you just grow the cells and then isolate the protein from the cells, or from the, sometimes it is actually inside the outside the cytosol, that can also happen. Or in many cases you can lyse the cell, separate the DNA, isolate the protein, and from the protein you purify and isolate your target protein. Okay. I will give some example here. Maybe right now I can give you that, suppose we know that in diabetes there are two kinds, one is insulin dependent, another is insulin independent. So, some people who are suffering from diabetes, they have to take insulin, that is the insulin dependent ones. Okay. And insulin, where from the insulin is obtained? Earlier it was obtained from the cow, the bovine. So, it was bovine insulin, that was the one which the diabetes patients were using. Okay. Now, later on it was found that it is not a very good practice to slaughter a cow and then isolate the isolate your insulin. First of all, it is not human insulin. There are some differences in the bovine insulin and human insulin, although there are good, there is good homology. Homology means that the sequence of the amino acids in the protein are very similar. Okay. The highest homology the bovine has, that is why the you isolated, the practice was that you isolate the insulin, and that bovine insulin was used by the diabetes patient suffering from diabetes which is insulin dependent. Later on it was found that if we can inscribe, suppose this is your insulin gene, And if that be the case, if you can take the insulin, human insulin gene and put it into as a recombinant DNA, put it inside a bacterial cell via this cloning technology, then what will happen? These cells, because it has got the insulin gene, so when it grows it will make the insulin, because it has got the insulin gene. And now, whatever insulin that are available in the market are all dependent on the are all recombinant insulin, okay, and which is the also it makes the human perfect human variation variation of the insulin. Okay. So, you see that this technique is so important, because if you want a protein, particular protein, if you are interested, usually the proteins are made in the cells in very small amount. Okay. If you want large amount of the proteins, then what your target will be, that you try to identify what is the gene, corresponding gene of that protein, and then take the gene and then put it as a recombinant DNA. That means, you attach it to the carrier DNA or the vector DNA and then put it inside a bacteria. If you are successful in that, then you can all the time you can get the protein, because as the bacteria grows you will get the it makes the protein, and that is the modern day technology for any laboratory who are doing proteomics or enzymology, protein related biology. Okay. Now, the questions to be asked here, that how do you make this recombinant DNA? That is number one. The second is basically I think that is the most important, because this recombinant DNA is basically what we are talking about, that you have a vector DNA and you attach your DNA of interest. Okay. How will you ensure that it has been attached? Because when you have this vector DNA, some of the vector DNA will remain as without any attachment from outside, okay, and some of the vector DNA will be attached to this external DNA. Okay. It is like very similar when I talked about the catalytic antibodies. There was this type of problem, that some cells, some immortals, but they only are made up of cancer cells, and some cells were there which are actually hybrid of the cancer cell and the and other foreign cells. Okay. So, that is the spleen cells which are making the antibodies. So, there also you have to select that which are the cells which are hybridoma and which are non-hybridoma cells. Okay. Here which cells are which DNAs are recombinant DNA and which DNA are only the vector DNA. So, you have to identify that. So, that is very important, and we will discuss that how it is done. Before that, let us talk about little bit of this restriction enzyme. Restriction enzyme, I told you that it recognizes palindromic sequence. It recognizes palindromic sequence, and this is the palindrome AAGCTT, and if you read from this side AAGGCTT, okay, same. So, that is a palindrome, a 6 base palindrome, and the enzyme that recognizes it, it is called HIND3. So, HIND3 recognizes this sequence and it cuts at this point and that point. So, between the two A's, the phosphodiester bond is cut, is clipped, and that produces this type of fragments, and this is what is called this, that means there is 5 prime protruding ends, okay, or overhang you can say. That means, a 5 prime overhang on this side and a 5 prime overhang on this side. Overhang means where there is no complementary strand present, there is no complementary strand present. So, that is, now this is what is called the sticky cuts. Sticky cuts means, again if you add these two, then because this A matches with T, this G matches with C. So, they will come and immediately they will. So, just if you think this is a block of wood kind of thing. So, then what you have is basically. Sorry. So, this part goes inside and then because there is perfect matching here. So, it goes inside and then you can again do the ligation. Okay. So, you need a ligase to ligate these two pieces, but this is what is called the sticky cuts, because there is a recognition point, there is a overhang which recognizes each other. So, similarly, there is this PST1, another enzyme which recognizes CTGCAG. Again, this is a palindrome CTGCAG. Okay. So, this also this gives a 3' overhang, 3' end is extended. Okay. And there is another type of cleavage that is called blunt cut, and that is done by EcoR5. EcoR5 recognizes GATATC. Again, this is a palindromic sequence, but it cuts, it is a blunt cut, it cuts straight away at this at the same point. Okay. And that gives what are called blunt ends. Okay. Now, it is easy to ligate these two, because they have a recognition arm, but here there is no recognition arm, because it is a blunt cut. But there are ways, we are not going into that details, but there are ways to also stick to this, to again join this together. Okay. But that is a much more complicated ones if you have blunt ends. On the other hand, people try to have sticky cuts, because sticky cuts are easy to join, just add the DNA ligase, they will join each other. So, this is what is the restriction enzymes. There are many restriction enzymes that have been isolated, and this restriction enzymes is present in bacteria, and it is believed that the bacteria has evolved these restriction enzymes in order to protect themselves from virus, from viral infection or other bacterial actually viral infection. So, that these restriction enzymes cuts this external DNA and chops the DNA apart, so that it cannot infect the bacteria. Okay. I think this is what restriction enzymes perform highly specific DNA cleavage reactions. Bacteria evolved mechanism to protect themselves from viral infections. Okay. So, if this is the viral one, the viral DNA, and if this is the host DNA. Now, you can always ask this question, that the sequence that is present in the virus, that has to be a palindromic sequence, and that has to be recognized by a restriction enzyme which is made by the host DNA, but the host DNA itself can have the same restriction same site which is recognized by the same restriction enzyme. So, how does it really protect itself from the self cleavage? So, what it does, one interesting point is mentioned here, that the bacteria labels the DNA, self DNA with the adenine with a methylation. So, it does a methylation of the adenine, and that actually indicates or gives an instruction to the endonuclease that this is my self DNA, do not cut it. So, better cut the other ones, that means the viral DNA which is not methylated, where the A's are normal A's. So, that is the bacterial way of identifying self and the external, okay, just by putting the methyl group to the host DNA, that means the bacterial DNA. Think now we are talking about this. The question is, how do we identify the recombinant the cells which are making recombinant DNA. Now, we did not talk about the vector yet, that we said that it is a piece of DNA which has replicability, which can replicate when it is put in a bacterial cell. Okay. So, this vector is usually what is found to be what are called plasmid DNA. Plasmid DNA is a circular DNA. This is called plasmid DNA. Plasmid DNA are circular DNAs are circular piece of DNA, and these are usually present. They are not usually, always present outside the. It is not present in the chromosomal DNA. It is not a part of the chromosomal DNA, but it is actually present within the cytosol. Suppose this is the bacterial cell, and suppose these are the chromosomal DNA, that means containing all the information to make the proteins. Okay. So, in addition to the chromosomal DNA, the bacteria has developed this what are called plasmid DNA. The plasmid DNA are the circular, they are circular DNAs, and they are not, they are extra chromosomal DNA, and bacteria evolved this plasmid DNA to confer resistance to different antibiotics. So, this is basically has evolved, it offers resistance or gives resistance capability. Resistance to what? Resistance to antibiotics, resistance capability. I can say antibiotic, I can add this word antibiotic resistance capability is provided in the plasmid. That means, the plasmid has a gene. If you go a little bit extrapolate this part, that the plasmids are developed because of to confer antibiotic resistance to the bacteria. That means, the plasmids are making some enzymes which are destroying our antibiotic before the antibiotic action is shown. Okay. The beautiful character of this means this is also very dangerous, because this is the mechanism by which bacteria developed resistance. But what was the problem? If bacteria had the resistance gene, it is there in the chromosome. The time taken from chromosome to express to the protein is much more than the plasmids. The plasmids can easily replicate, they have very good replicability, and I said that is why they are vector, and they can also very quickly infect other cells which do not have these plasmids. So, there could be cross infection of this plasmid to the bacterial cell which does not have this plasmid. Okay. And that is how the resistance can be spread into the entire bacterial population very quickly, because they do not you do not have to unwind the entire chromosomal DNA in order to get that resistance gene. It is actually present in the plasmid. So, plasmids are having these characteristics, that they have antibacterial resistance genes present there, and also and that could be there could be many genes. One is acting against tetracycline, another is acting against penicillin. Okay. There are many antibiotics we have class. So, there may be different antibiotic genes, resistance genes present in the plasmid, and they can cross infect. So, they can also easily multiply. Okay. So, in this technique, what is, I can show it here, that basically what happens here is that you take a plasmid which has got, now plasmids can be synthetic also, means now people, there are different plasmids that are available which are made synthetically, synthetic plasmids containing different genes expressing for different proteins. Okay. Like let us take this one, this is called what is called PBr322. This PBr322 plasmid has two genes, one gene confers resistance to the ampicillin, ampicillin is a penicillin, and there is another gene which confers resistance to tetracycline. So, this is my starting vector. Okay. So, this vector is PBr322. If you do not remember the name, follow the principle. The principle is that it should have, the plasmid should have two identifying to different kinds of genes expressing for two different proteins, and that gives some the resistance to two different antibiotics. But there could be plasmids where there could be one antibiotic resistance gene, another could be a gene for a protein which gives color. So, that is also possible, means there could be different variations of plasmids. So, this one, let us talk about this one, this has got two types of resistance, tetracycline and ampicillin. Now, what has been found that this ampicillin resistance gene, inside there is a site which is a restriction site for this enzyme PST1 restriction endonuclease. So, that means there is a restriction site, these sites where the restriction enzyme works are called restriction sites. So, this gene has a restriction site inside the ampicillin resistance gene. Okay. So, if you now treat this vector with PST1 restriction enzyme, so what will happen? This will cut and this gives a sticky cut. I told you last time that PST1 gives a sticky cut, HIN3 gives a sticky cut, ECO R5 that gives a blunt cut, but this is a sticky cut. So, that cuts it into this type of pieces open. This is the ring is not closed. Okay. Now, if you have the foreign DNA, suppose I have the foreign DNA like this and I am interested only in this part. Now, if there is a restriction site here, suppose there is a restriction site here and a restriction site here, these are recognized suppose by PST1, Okay. So, then what will happen? Then you will have pieces where the ends, the restriction, this after treating with the restriction site of the foreign DNA, remember this part should not be disturbed at all. So, you are cutting at the little bit far on this side and little bit far on this side, and then this will have similar kind of sticky ends. So, this will be the picture, and then when you ligate, that means this part now can ligate with the protruding part or the overhang part of this end, and the other end can ligate with the overhang part of the vector DNA, which is already treated with the same restriction enzyme. So, basically the principle is that you should use the same restriction enzyme to cut your external DNA and also to cut the plasmid, and the plasmid is cut in such a way that your ampicillin or any of the antibiotic resistance, the marker, these are called marker genes, and these genes are cut, one of the gene is cut inside. So, that this, if you now express it in the bacteria, so because these two are not joined, so this is a disjointed gene. So, that will not express the enzyme that destroys the penicillin, ampicillin. Okay. So, that is the reason that why you cut inside the ampicillin resistance gene. Now, you add this part, this is your foreign DNA with the same sticky ends, and this is your vector DNA with the same sticky ends. Now, when you add DNA ligase, what will happen? Some of the vector will just again bind to each other, because they can recognize each other. So, some of the, this is the recombinant DNA. Now, some of the vectors are they not taking any external DNA, but this external DNA can recognize that part. So, now some of the vector, I can show it here. Now, some of the vector will remain like that, remain as earlier. So, they just join with each other again, but some of the vector can take this external piece of DNA. Suppose this is the external piece of DNA, and then you complete the other part. So, that means you have a, now you have a this is recombinant DNA, rDNA, DNA, and this is your just a vector DNA. So, you have now two types of DNA, one is the normal earlier one, that means this one, and the other one is a vector DNA, is a recombinant DNA. The difference between the two is that, see, remember this was your ampicillin resistance gene and you have cut inside, and then you have put a foreign DNA in the rDNA, recombinant DNA. So, this recombinant DNA lacks the resistance against ampicillin. ampicillin, okay, because your gene is disjointed, because this is the gene, in between there is something foreign DNA. So, now what you do, you take this all this recombinant DNA plus the original vector DNA and put it inside. That is not difficult. You can transform E. coli cells, that is a particular type of bacterial cells, gram negative bacteria, and you put this DNA. Okay. Some cells will have the vector DNA, some cells will have the recombinant DNA. Okay. Usually one vector, one plasmid in one cell. Okay. Now, you grow this in a agar plate where the bacteria goes, can be cultured in a petri dish, and this is containing tetracycline. So, when the bacterial cell, when this population is transferred here and they are allowed to grow, but the medium you have already added tetracycline. If you have added tetracycline, remember tetracycline gene was not touched at all. So, it was there in the vector DNA and it was there in the recombinant DNA both. So, there will be bacterial colonies everywhere. Okay. All the two types of bacterial cells, one containing the vector DNA and the other containing the recombinant DNA, both will grow here. Now, what you do, you take another plate and two agar plates, one containing ampicillin, and see now you take two plates, one plate containing tetracycline and the other plate containing ampicillin and tetracycline. So, this is ampicillin and tetracycline both, and the other is only tetracycline. Okay. Tetracycline, these are both are antibiotics. So, now what will happen to matching positions? That means, if you are taking a bacterial colony from here, you are taking it at the same position, that means, you make a grid kind of thing. You make a grid and then according to the xy coordinate, like this bacterial colony is, you place that here. Okay. And also you have the same, you make the grid and then matching positions, that means, the colonies are transferred from here to the same xy coordinate, the matching positions you have to transfer. Okay. Now, what will happen here, because you have here tetracycline, only tetracycline. So, what will happen, that when you transfer it, so tetracycline resistance means everything will grow again, just to cross check, everything will grow, because both the vector DNA and the recombinant DNA has the tetracycline gene. But when you develop it into ampicillin plus tetracycline, then only the colonies, only the bacterial cells which have got only the vector DNA, because it is the vector DNA which confers resistance to both ampicillin and tetracycline. So, the colonies that are growing here is containing only vector DNA. So, now you can realize that this colony is not present here, that means, it is not growing. So, that means, this is a recombinant, this colony must be having a rDNA. So, by this technique you can compare at different points. So, and then like this one, it is shown here, this is not there. So, if it is not there, that means, this colony must be a recombinant DNA. So, that is how now you take the colony and then grow in another media, grow in a suitable medium, and then you get the copies of the, when the bacteria grow. So, you get copies of the recombinant DNA. As I told you, now it is, if you are interested only in the DNA, then isolate the DNA, but if you are interested in the protein that this rDNA is making, then you isolate the protein. But in both the cases, you have to lyse the bacterial cells and then separate the DNA, nucleic acids, and then isolate the protein of interest. So, this is the I told you, this is kind of a semi in vitro, because initially the reactions with the restriction enzymes are done in vitro, and then that is transformed into the bacterial machinery, and then after that it is in vitro, in vivo technique. Okay. So, this is one way, it is a beautiful way. Now, many of the problems have been solved, that recombinant DNA technology can generate or give copies of DNA as well as the proteins, that are the most interesting aspect that carries out all the reactions that you want. Okay. So, a very useful technique which was developed in the, I think, the early 1980s, this recombinant DNA technology was made available, and now it has become a routine technique in any molecular biology lab. Okay. The next session we will discuss the entirely in vitro method, and that is called the polymerase chain reaction. So, the second technique we first discussed, now we will go back to the first technique, which is the polymerase chain reaction. Thank you.